tobacco consumption is still increasing. Depending on estimate, at least 15 billion cigarettes are consumed every day. And as you know, there are about 4,000 compounds in tobacco smoking, and greater than 70 of them are established carcinogens. And this map shows the number of deaths by tobacco smoking in 2016 all over the world. And you can notice that this is not corrected for population size, and so the countries with the high rate of death are the countries that are highly populated. But the point of this one is just to show that if you quickly take a look, it's a global problem, and if you put this together, you get that at least 10, 12 million people every year die of tobacco-related disease. And when you're looking at the impact of tobacco smoking health-related, it's not just cancer, as indicated by the tissues in red, but you impact pretty much every single tissue in the body, and it could be from serious complications like heart attack to more cosmetical, like stinking of tobacco smoke, teeth problem, and so on. And as you well know, it's estimated that tobacco smoking is responsible for at least 90% of all lung cancers and for 75% of all chronic bronchitis and emphysema. And what we're going to focus today is the evidence that we have on the impact of tobacco smoking on reproduction. When you think about an abnormal reproductive effect, you need to keep in mind that the result of the abnormal reproductive outcome may go back at least a couple of generations. And we're going to hear more about this one also from the speakers later. But basically, you need to consider the fact that the germ cell that produced that abnormal outcome may had a long history that goes back to being a primordial germ cell in the grandmother of the child with the abnormalities. And this is just a very schematic representation of landmarks that are happening for both maternal germ cells and paternal germ cells during the in utero exposure after the birth of the parents. And every of these landmarks may be susceptible to an induction of genetic or an epigenetic alterations that will contribute to the abnormal reproductive outcomes. So you need to consider as a whole the history of that family to really understand the cause of that reproductive outcome. In addition, when you start talking about how you get from an exposure to an abnormal reproductive outcome, there are multiple steps along the way. And again, this is a schematic representation of what you need to consider. As Kerry pointed out, you need to understand the exposure, the external exposure, because as he said, if there is not an exposure, there is no risk. But also, it's not sufficient to just understand the external exposure itself, but also how much of that exposure actually reaches the target tissue, in this case, the testes or the ovary. And then, based on the internal dose, you need to also have to understanding how much of that internal dose you need to start affecting some important cellular processes or molecules, and how that triggers an early biological response from the system that may be able to compensate for that exposure. And you can distinguish between early response, for example, activating a cell cycle control, for example, recognizing that you have been hit, to intermediate biological effects, and that the end result is that if you're not being able to properly take care of that insult, you will end up with an abnormal reproductive outcomes. And all of these steps can be modulated by host factors that can be including genetic susceptibility that may contribute to how much or how fast along this path you go. And in addition, something that I will not have time to go over on, there is also the fact that when we're talking about paternally transmitted effect, it's also very important what is happening in the egg in the first few hours after fertilization and how the egg is able or not to deal with whatever the sperm bring into the egg. And that can have a profound impact on whether you're going to have a genetic defect that is transmitted or that is taken care of. As I said, I'm going to have a couple of slides on female germ cells, and this is just summarizing some of the evidence we have of the impact of tobacco smoking on the female germline. We know that maternal smoking is associated with a reduction in the number of oocytes that are retrieved when the women are going for in vitro fertilization clinics. There is a dose-related decrease at the age of menopause, at least a 2-month delay in natural conceptions, and also a significant increase of spontaneous abortions that affects both women who are undergoing IVF, but also natural fertilization. So there are fundamental and critical impacts of maternal smoking on reproduction. When we are thinking about the effects in male germ cells, one thing to keep in mind is that the type of damage that can be induced by tobacco smoking or any exposure, it's dependent on the stage that the stage susceptibility of germ cells and how they respond. And this one is a schematic representations of spermatogenesis. You can divide them spermatogenesis in a mitotic phase, a meiotic phase, and a post-meiotic phase, and the different cell types that you find during spermatogenesis. This is a more realistic representation of the structure of the seminiferous tubules. And so here you have the tube, the basal lamina, and then going from the basal the tubules into the luminal side, and the more the spermatogonia are toward the base, and then as they progress through spermatogenesis, they move toward the luminal. And here is the one important component of the blood-testis barrier, the tight junction between two Sertoli cells. And here you can see that the spermatogonia is on the open side of the barrier. And what is really needs to be kept in mind, especially when we're talking about genetic damage, is the fact that DNA synthesis is confined mostly to the mitotic phase of spermatogenesis. And in addition, that toward the end of spermatogenesis, the ability to repair DNA damage goes away. And so this one becomes a sensitive window for the induction of DNA adducts, strand breaks, and as Jacquetta mentioned, also protamine adducts that may result in chromosomal damage after fertilization. And if you're looking at point mutations, the target cell is more spermatogonia, and if you're looking at aneuploidy or recurrent copy number variants, then the spermatocytes is the cell target more sensitive to the exposure. I'm going to now quickly go through the evidence that we have for tobacco smoking inducing genetic effects in male germ cells, and these are basically summarized here. And I will show you data showing that paternal smoking is associated with impaired fertility and sperm DNA damage, alterations in chromosome numbers in sperm, tandem repeat mutations, and also congenital malformations and cancer in the offspring. And this one is nicely summarized by a review that we published a couple of years ago, and Carole mentioned that, and McGill was instrumental in putting all this data in summarizing this data. And so we start looking at the effects of tobacco smoking on fertility and DNA damage in sperm. and these are the types of parameters that there is data available, the specific endpoint that was measured, the species where the endpoint was measured, the number of studies that are published, how many studies had a significant effect of smoking, and the effect size for those studies. And you can see that there are plenty of studies confirming that depending on level of smoking, there is an impact on fertility and DNA damage. And the other thing to point out, that especially for some of these sperm assays, we also have quite a lot data in humans. So we don't have to rely just on animal models, but we can really look at effect on human germ cells. There is also, want to briefly go over this study, because this is one of the few studies where multiple endpoints were analyzed and not just a single endpoint. And this is a study that we did a few years back when I was still at Lawrence Berkeley, in which we exposed male mice to not only mainstream smoke, but also to side stream smoke. There was a question earlier this morning about the impact on not just first-hand, but second-hand and also third-hand smoke. And then we measured different endpoints related to both DNA, sperm DNA integrity, but also an effect on reproduction, looking specifically at fertilization rate and also embryonic development, and different, the mice who were exposed for 2 weeks to either mainstream or side stream, and then analyzed both for the sperm and reproduction, but then we also waited at least 42 days to look at effects in mitosis. And you can see that we saw significant effect in most of the endpoints that we analyzed. And interestingly, we also see some difference in the response to mainstream and side stream smoke for some of these endpoints. So that side stream smoke is not exactly equivalent to mainstream smoking. The other endpoint for which there is good data demonstrating an effect of tobacco smoking on male germ cells is the data demonstrating the impact of aneuploidy. And interestingly, these are all human studies in which they analyzed the presence of numerical abnormalities in the sperm. And you can see that in 7 of those 9 cases, they found a significant effect, and the effect was from 1.2 to 4-fold increase. And I'm giving you this number because when you look at the effect of chemotherapy in human sperm, you find a 5-fold increase in numerical abnormalities. So you can see some of these studies, the effect of smoking was almost equivalent to the effect of chemotherapy when you receive high doses of strong genotoxic agents. Then there is also evidence that tobacco smoking is induced with induction of tandem repeat mutation in both sperm and children. Again, it's another endpoint where there is data in both humans and rodents. The number of studies are small, but they were all, pretty much all studies found a significant effect, and specifically mouse studies showed the smoking increased tandem repeat mutation in sperm by 2-fold, and the studies in humans was also able to demonstrate not only an effect of smoking, but that there was also a dose-related effect. So fathers that smoked more had transmitted more tandem repeat mutations to their children. And this one, it's the other slide summarizing studies that have looked at childhood cancer, congenital malformation, perinatal mortality, spontaneous abortion, again, all done in humans. And you can see that a significant fraction of these studies found a significant effect, and the overall results are summarized here. Childhood cancer is associated, these are the type of childhood cancer that have been associated with not only paternal but also maternal smoking, all a long list of different congenital malformations, and also pregnancy loss and perinatal mortality. And still continuing the association between tobacco smoking and cancer in the offspring, these are the statements that are included in the last IARC monograph that evaluated personal habits and combustions. And I think there was 2012, David? 2000? Well, recently. And the IARC panel concluded that there is a causal link between parental smoking and childhood cancer. We went over 4 recent studies that show that children born to parents who smoke, father, mother, or both, are at significantly higher risk of this hepatoblastoma cancer, which is quite a rare cancer. And then some evidence that only paternal smoking as well was associated with an increase in cancer, and that there is specifically childhood leukemia. This meta-analysis and association between paternal smoking before pregnancy and the risk of having childhood leukemia. So the evidence that paternal smoking can contribute to cancer in the offspring, it's quite solid, I think. We're also starting to get some data showing that the effects, there are also some reproductive effects that are affecting not just the smoker, but his offspring. There are many studies in humans that now have shown that maternal smoking during pregnancy reduced sperm count in sons, but now we're starting having the evidence that the same is happening when the father is the smoker. And there was this initial study by Jonathan, who's actually in the room today, showed that men of smoking fathers have a 41% lower sperm concentration with respect to the men of non-smoking fathers. And there is now a paper that it is not published yet, but there is a report of a study from the Danish National Birth Cohort that analyzing over 800 cases and confirmed that paternal smoking is associated with lower total sperm count and sperm concentration in their children. So I think now we're starting to have some very solid evidence that you are impacting not just your reproductive chance, but also the chance of your children to reproduce. And so if we put that together, this is my personal interpretations of the level of evidence that we have for tobacco smoking inducing these types of effects. So if you're looking at impaired fertility and sperm damage, I think we have sufficient evidence that it reduces sperm function. There is strong evidence that increases DNA adducts and DNA damage. There is sufficient evidence that induces aneuploidy in sperm. There is sufficient evidence that it induces tandem repeat mutations in sperm. I consider the evidence in children weak because it's only one study, so it would be nice to have additional study confirming that. And then I think that there is strong evidence for paternal tobacco smoking inducing congenital abnormalities, and sufficient evidence for both cancer in the offspring and reduced sperm count in the children of smokers. I'm not going to spend time on this one. Carole went over that, but this is just to remind that even a small increase in mutation may have a large impact at the population side. And so this is the slide that's trying to put everything together with respect to genetic damage. And so we have now conclusive evidence that paternal smoking may result either in reducing sperm count or sperm that have some DNA damage that then is transmitted to the embryo, and that DNA damage can have either result in pregnancy loss depending on the mutation that is transmitted, but more importantly result in a baby that's born with genomic alterations, potentially susceptible to genomic instability, genetic disease, and reduced fertility. And I think that on top of this, we can probably create a similar depiction for epigenetic effects, and because probably you can end up with the same types of endpoints at the end. So what do we need to do moving forward? I think that clearly the hanging fruit is to use these new genomic tools to establish that smoking is clearly a human germ cell mutagen by increasing the number of mutations in the offspring of heavy smokers. It would be really important to quantify the effects in spermatogonal stem cells, because these are the cells that are present throughout the life of the individual, and so if you affect them, even if you then stop smoking, the damage is done. It's now there's the ability to evaluate the mutation signatures that can give you confidence that an exposure is happening and the type of exposure that is happening by looking at the pattern of mutation. And finally, as we said repeatedly, we need also to look at multi-generation studies to understand how smoking during pregnancy affects the children and the grandchildren of the exposed parents.